All right, you are on page 77 in your binder. We're going to go through enzymes. We are moving forward and skipped over some things, and then we're going to go back to those things later on. We just had an issue with one of the balances in one of the labs. So we're moving to enzymes, and then we'll go back to cell membrane. After you watch this video, make sure that you have the notes on page 77, 78, and the top of 79 filled out, uploaded to Teams before the next class period. Also make sure you understand the topics that are discussed because this takes the place of an in-class lecture. It is a familiar topic. It should be a familiar topic from cell bio. There's really not going to be anything that new that you're going to see today, like at all. Next week, you're going to have a quiz over chapter 4 and this, so make sure you understand it. I will ask you at the beginning of the next class period, do you have any questions? And if nobody has any questions, I'm going to assume everyone understood it. All right, so you guys know what enzymes are. We talked about them when we talked about proteins. So the first thing I need you to know is that an enzyme is a protein. Make sure you pause the video anytime you need to write something down. I'm not going to do pauses during the video, so just pause it. Enzymes are proteins. Enzymes are going to be involved in metabolism. Metabolism is the totality of an organism's chemical reactions, and so those chemical reactions are going to occur because of the help of enzymes, which are also known as catalysts. They're going to speed up the rate of reactions. So pause if you need to write that down. Metabolism is also the management of materials and energy resources of a cell. And so enzymes are going to be involved in that. So the picture that you see up here is A is being converted to B with the help of enzyme 1. B is being converted into C with the help of enzyme 2. C is being converted into D with the help of enzyme 3. So all of these chemical conversions and these chemical reactions are happening because of the help of enzymes. An enzyme can be defined as a catalyst, which is a substance that can change the rate of a reaction without being altered in the process itself. So something you're going to see when we do the lab this week is that an enzyme is unchanged. So not being altered. Write the word unchanged under that. And an enzyme is what's considered a biological catalyst. All right. Here is an example of sucrose, which is a disaccharide being broken into glucose and fructose with the help of sucrase. I hope you would recognize this reaction as being a hydrolysis reaction. So sucrase is basically inserting water into that bond and breaking that bond. An enzyme is going to speed up a metabolic reaction by lowering this thing called activation energy. And activation energy is the energy needed to start a reaction. So you can think of activation energy. Here's my analogy. If my child is sitting on the top of a slide, the natural tendency of the universe and gravity is for her to go down the slide, right? But she's just sitting on top of the slide, not going down. I need to give her a little push at the top. And then it turns into a spontaneous downward motion because of gravity. So an activation energy would be the little push that's given at the very beginning of a chemical reaction. And so that push that's needed, an enzyme is going to lower the energy that's required for that first initial push for a chemical reaction to happen. I need you to recognize this picture right here. This picture is showing a spontaneous reaction. The black is the energy required for, you see this hump? That's the activation energy required to get the reaction started. The red is the same reaction with the help of an enzyme. So I need you to recognize this picture. Ignore delta G. We're not going to deal with Gibbs free energy in this class. But I need you to recognize this picture as an enzyme is lowering the activation energy, the energy required for a reaction to begin. All right, next page. An enzyme has a specific folding because it's a protein. It also has a specific fit for a substrate. So the reactant that an enzyme acts on is called the enzyme's substrate. The enzyme binds to the substrate, forming an enzyme substrate complex. And the active site is the place where the enzyme and the substrate bind. So pause if you need to write that down. Here is a diagram. Oh, it's supposed to show a, dang it, it's supposed to show an animation. Well, that stinks. Okay, the yellow thing is your enzyme. All right. But does this work? My pen. All right, the yellow thing is the enzyme. This place right here would be, sorry, the active site. This is the active site. And then your substrate would be what I have in red here. All right, so the enzyme is yellow, the substrate is in red, the active site is the place where the substrate fits. How do I erase this now? Eraser. Woo, okay, I learned something new. Cool. All right. So go back and pause if you need to. This is right here showing you the steps. The steps. You're going to need to write all of these steps down. So you're going to probably need to pause. Step 1, the substrate enters the active site. Step 2, the substrates are held in the active site by weak interactions. What is an example of a weak interaction? Van der Waals. Step 3, the active site lowers the EA. What is EA? It's activation energy. Activation energy. So the active site is lowering the activation energy of the chemical reaction, speeding it up. Step 4, the substrates are converted into product. Step 5, the products are released. And step 6, the active site is now available for new substrates to come in. You're going to need to write down all 6 of those steps. So pause the video if you need to, because I'm moving on. There is something called induced fit. Induced fit is the enzyme fits snugly around the substrate. So not only does the substrate fit into the active site, but the enzyme will kind of wrap itself around it, and that's called induced fit. And it's like a clasping handshake. So definitely know that vocabulary word. The substrate comes into the active site, it fits in, but then the enzyme kind of gives it a big hug right here. That's called induced fit. All right? An enzyme's activity can be affected by, and you already know this. You already know this because of that protein, that egg white lab that we did. If an enzyme is a protein, then anything that affects a protein can affect an enzyme. So on the bottom of page 78, where it says what 3 things can affect an enzyme's activity, you're going to write down these 3 things: temperature, pH, and chemicals. The graph right here, so this is on the top of page 79. So pause to write those 3 things down because I want to talk about these graphs. It's the last thing I'm going to talk about in this video, and it's really important. These 2 graphs on the top of page 79 is showing you a black curve and a red curve. The black curve is showing you the optimal, this top graph here. So make sure you're paying attention to this. The black curve is showing you the rate of reaction as it relates to temperature. The black is showing you a human enzyme. The red is showing you an enzyme for a thermophilic bacteria. The top of the curve is showing you, and this is an important word, optimal. That means the best. That means the rate is the fastest at this point. So the optimal temperature for an enzyme in a human is 37 degrees Celsius. And that should make sense because that's the temperature of the human body. Look at this red curve over here. Look at my cursor. The optimal temperature for an enzyme for a thermophilic bacteria is 77 degrees Celsius. And that should make sense because a thermophilic bacteria is functioning and living in something really, really hot. All right, come down to the bottom here. Now this is given 2 enzymes found in humans. The first one is pepsin, which is an enzyme found in the stomach. The second one is trypsin, which is an enzyme found in the small intestine. Both of these enzymes actually digest proteins, so digest meat when you eat them. They're just located in 2 different places. So these curves right here, black one for pepsin, red one for trypsin, both are enzymes found in the human digestive tract. But pepsin is in the stomach, so its optimal pH is 2. Trypsin is found in the small intestine. Its optimal pH is 8. And those are the pHs of the stomach, and that is the pH of the intestine. So those are the optimal pHs for those 2 enzymes. All right? So if it's found in the stomach, its optimal pH would be 2. If it's found in the intestine, its optimal pH would be 8. Those are the pHs at which these 2 enzymes operate the fastest. Okay, I made it to the 10-minute mark. Make sure you have those notes uploaded to Teams. Thanks.